When we look at advanced pancreatic and non-small cell lung cancers, we find that many of their most aggressive forms are driven by a single genetic error. This data illustrates the footprint of a specific mutation known as KRAS G12D. On the left, it acts as the primary engine for about 40% of pancreatic ductal adenocarcinomas. On the right, it appears in roughly 5% of non-small cell lung cancers. Despite being common and lethal, the KRAS G12D protein has historically been considered entirely immune to modern medical intervention. Patients carrying this specific genetic mutation have typically been left with few targeted treatment options, leaving a gap in modern oncology that researchers have struggled to fill for decades. Traditional targeted therapies work by physically binding to a deep pocket on a cancer protein to block its activity. But the KRAS G12D protein lacks this keyhole. It's a smooth structure where the geometric drug molecule simply bounces right off. This structure makes the protein essentially a fortress. If a drug cannot physically bind to it, it cannot block the cancer's growth signals. As long as pharmacology relies exclusively on this blocking mechanism, mutations like KRAS G12D remain scientifically undruggable. That brings us to an international Phase 1 clinical trial testing a different investigational drug called Setidegrasib. This trial, published in the New England Journal of Medicine, was conducted across 28 centers in 5 countries to evaluate the drug's safety and early activity. Setidegrasib operates on a different logic. It is not an inhibitor, it is what's known as a protein degrader. This class of drug abandons the attempt to stall the cancer protein's activity. Instead of trying to put a cap on a broken machine, Setidegrasib is designed to dismantle the machine entirely. Inside a cell, Setidegrasib bypasses the need for a deep keyhole by chemically tagging the abnormal KRAS G12D protein. This tag signals the cell's waste disposal system to recognize the cancer-driving protein as garbage, breaking it down and removing it. Laboratory analyses showed that this process successfully lowered the physical amount of tumor DNA circulating in the patient's blood. By removing the protein entirely, rather than merely capping it, the drug takes away a key tool the cancer cell needs to survive. To see how this biological action translates to patients, researchers looked at 66 people who received a recommended 600 mg weekly dose. In non-small cell lung cancer, 36% experienced tumor shrinkage, with a median of 8.3 months before progression. For pancreatic ductal adenocarcinoma, 24% saw shrinkage, and overall survival reached 10.3 months in heavily pretreated patients. While the trial noted some infusion-related reactions like rash and nausea, the treatment was generally well tolerated and manageable with standard care. These are early signals, but in cancers where targeted options have been scarce, they provide concrete evidence that protein degradation is a viable path for further study. This trial suggests that we may be able to move past the structural limitations that once made KRAS G12D undruggable. Instead of relying on temporary roadblocks, researchers are testing ways to force the cancer's architecture to dismantle itself. This work was led by an international team, including investigators from UCLA Health, shown here, and Memorial Sloan Kettering. If confirmed in later trials, it could mark a broader shift in cancer therapy, moving from drugs that inhibit cancer proteins to drugs that eliminate them.